let's study about targeted cancer therapy. In case we have big chunk of tumors, maybe first the target would be reduce the size of this tumor. So that means aggressively attack the tumor by adding a lot of toxic materials. That is so-called chemotherapy. The situation may be similar to this kind of a full-scale war. So in this case, to attack the tumor, we send in maybe the very vigorous and toxic chemicals like missile and fires. Then in this situation, a lot of side effects such as the toxicity to other fast-growing cells in our body and a lot of other problems can occur together. If there is other method like this, so let's say the enemy's headquarters and headquarters send out signal to the battlefield, and rather than attacking directly the military forces, maybe destroying this headquarters may be a better idea. In this case, maybe the target is not directly the military force, maybe the signaling the bodies. So if you can cut those signals from headquarters to battlefield, maybe this is a more efficient way to attack the enemy's strategy. So here is the very complicated biochemical pathways. It looks like quite complicated. And in a sense, this is kind of almost everything we know about our the body in terms of chemical reactions. Now, if you wanted to attack the tumor vigorously, that's like attacking the chunk by chunk and destroying those the cancer's biochemical machineries. But this would generate a lot of a problem. Then, if you wanted to attack that specific signaling, then maybe we can choose one specific target. So rather than destroying the whole body, just kill only one protein or the enzyme, then that would be a more specific approach. Then, comparing to firing or sending missile, maybe this is like a sniper the operation, just to kill only one specific the important the target. Then that method would be quite specific and less toxic. And this approach is called as a targeted cancer therapy. Now, let's think about the mechanism of a signal transduction. When drug molecule needs to enter the body, they need to penetrate this cell membrane. Cell membrane is the boundary of life, and then they protect the inside from outside, and then prevent a lot of materials free entry to the cells. So that's why there are serious considering we need to take for developing the efficient drug in terms of the overcoming the permeability problem by improving their pharmacokinetic parameters. Signaling also has similar situations. If the molecule can enter cross this cell membrane and reach the target and then generate a response, then that would be straightforward. And if this is a signaling molecule, then this kind of a signal transfer we call signal transduction. But in many cases, the signaling molecule, in this case let's call ligand, this ligand cannot penetrate directly through the cell membrane. Then it needs to take indirect method. So in this case, there's one possibility is there are the receptors on the cell surface, and then ligand bind to receptor, and indirectly this receptor relay those signal of this ligand inside the body, and then that signal is relayed and eventually reach the final target, and this whole process is called signal transduction. Now let's take specific example. So in this case, if the ligand coming to the surface of the cell and bind to the receptor, and then this ligand is called as the first messenger. There is a first body which carries the signal. After this receptor is activated and that transfers that signal to G protein, and then enzymes are called PLC, and then PLC hydrolyze PIP2 into the IP3. This is inositol trisphosphate. Then in this case, this IP3 is the second messenger of the first messenger. IP3 can mobilize the calcium inside the cell, then that is the next signal. Then in this case, this calcium is called as a third the messenger. Here, PIP2 can generate IP3, but at the same time, it can also generate another product, DAG. Then DAG is also can be another kind of a second messengers. So in this example, the first messenger cannot penetrate the cell membrane, but it binds to the receptor on the surface. But that signal is transferred and then eventually generate the second messenger and third messengers. And these messengers generate the responses inside the body. Another big class of a signal transduction pathway is through kinase activation. When the ligand binds to the receptor, this activated the receptor can generate dimerization of its effector molecule. In many cases, it's a kinase. Once they become this dimer, then it is activated, and then it can recruit the substrate. Then it can phosphorylate its substrate. Kinase is the enzyme which transfers the phosphate from ATP to the substrate. Once the target is phosphorylated, then it can be dimerized, and then this dimerized form is activated form of the next signaling molecule, and in many cases they enter the nucleus and act as transcription factor. Transcription factor is binding to the promoter of a specific gene, and then it can activate this gene's transcription and eventually generate the expression of this gene and make the proteins. So this is the end point of this kinase pathway of signaling the pathways.